Nanomedicines, like the mRNA vaccines, are also used to deliver drugs, mainly chemotherapeutic drugs for cancer treatment. However, different patient tumors may accumulate these medicines at different levels, from quite a lot to almost nothing, affecting how well the treatment works eventually. Currently, there's no reliable and broadly affordable way to predict before starting treatment how much of the nanomedicine will reach the tumor. A stratification of patients can enable personalized therapies and ensure that patients receive a treatment which has a higher chance to work efficiently. In our research, we identified specific biomarkers, such as blood vessels and macrophages in the tumor, that can predict how well a tumor will take up nanomedicines. We confirmed these findings in both animal models and human samples. We addressed a key challenge in the field: the need for a simple and widely available way to identify which patients are most likely to benefit from nanomedicine treatments. Instead of using cumbersome imaging techniques like nuclear imaging, we used standard tissue staining methods commonly available in pathology that are also typically used for tumor diagnosis. This makes our approach more practical and accessible for real-world clinical use. Additionally, our work was strengthened by long-lasting collaborations with leading scientists and institutions, which brought together a wealth of expertise and resources, namely AstraZeneca, the IMC in Prague, the University of Utrecht, and the Pathology and Medical Clinic 3 of our Uniklinik. By using biomarkers like blood vessels and macrophages, we will be able to help doctors make better decisions about using nanomedicine-based cancer treatments. This could lead to more personalized and effective therapies for patients, ensuring that only those likely to benefit receive these treatments. Our findings could also help researchers and pharmaceutical companies to select the right patients for clinical trials, improving the development and clinical translation of new therapies. We are continuing to explore other biomarkers, such as serum proteins, to further improve our ability to predict how well tumors will respond to nanomedicines. In the long term, we hope to collaborate with clinical partners to further validate our findings in clinical trials, which could potentially benefit patients by providing more targeted and effective therapies. If I could turn back time, I would encourage myself to start working on the implementation of human samples at an earlier point in time. By using the CIO ABCD network, we could have tried to get access to even more patient samples and maybe also include patients who were treated with nanomedicines, which we are trying to do right now. Our research has been supported by several funding agencies over the years, first by the German Research Foundation DFG from 2011 on. This study was also supported by the European Research Council ERC, the European Union, AstraZeneca's postdoc scheme and the DFG through a research training group.